Welcome to the Heart of Healthcare podcast.
Hello, listeners, and welcome back to the Heart of Healthcare podcast. I am your co-host, Steve Krause, and today I'm really excited to have one of the true pioneers and leaders in the diagnostic space over the last 2 decades, Helmy Eltoukhy. Helmy is an American scientist and business person. He co-founded actually 2 very successful startups, which I want to talk about, Avantome and Guardant Health. Guardant Health, he literally started and has grown to, I think today it's a $17 billion market cap company. Is that, I believe, plus or minus?
Depends on the day.
Depends on the day, Helmy, of course. But congratulations. And for those who don't know, Guardant Health is a pioneer in non-invasive liquid biopsy, which is a really interesting, growing, important space in diagnostics. And so excited to have you on the podcast, Helmy. Thanks for joining us.
Oh, thank you for having me.
Yeah, we talk a lot actually on the Heart of Healthcare about healthcare IT and services, obviously AI these days, public policy. And from time to time, we have a few diagnostics guests. So I'm really excited to cover this really important broad field. But I want to start. You, I think, were a quant trader early in your career, but then went and did a PhD at Stanford, worked partly on the Human Genome Project. Then I noted you founded one startup called Avantome, which actually you sold to Illumina and is a big part of Illumina's business. And then from there went and founded, we mentioned Guardant. So I guess the question is, how does someone who goes to undergrad, works in quant trading, then gets his PhD, how do you end up being the co-CEO of one of the most successful companies in the diagnostic space?
Yeah. It's actually kind of an interesting story. I grew up in the Valley here. My dad was an entrepreneur. He started a couple of companies in the hard drive space. And then he started a hedge fund, actually quant funds. And I kind of used to work summers and work on models and so on for futures trading. I was always like a big math nerd. I was a mathlete. I did a bunch of math competitions growing up. And so I went in electrical engineering. I remember looking back at some of my kindergarten or first grade sort of writings and saying I wanted to be a neurosurgeon, wanted to help people. And so I always had that as a sort of underlying driving force, getting to medicine, going in that route. But when I went to Stanford in undergrad, looked and said, "Oh, should I be pre-med?" And I said, "Oh, it takes 15 years to get all the degrees that you need, residency, fellowship." And I was like, I don't have the time.
That's the time it takes to build a startup.
Yeah, exactly. So I said, let's go into electrical engineering. So I did that for a number of years. I did an undergrad. I did a minor in computer science. I did a master's in engineering. And then I kind of found out that doing straight electrical engineering was pretty hard. I was looking at these papers that the semiconductor companies were sort of publishing, and they had 50 authors on them. They're using $1 billion fabs to do this. And I was how am I going to compete as a lowly PhD student and actually move the standard of care, the kind of, state of the art? And so this project came along that was essentially combining semiconductors with DNA sequencing.
What year is this? Tell me.
This is probably the year 2000.
Okay, so early on-ish.
Yeah, early on-ish. It was, they were still finishing up the Human Genome Project. And so that's how I sort of began this detour into biology and biochemistry. Was we started working with the Stanford Genome Technology Center, really on the tail end of the Human Genome Project. I think it was mostly finished in 2001, and I had to learn biology, which I hadn't taken since high school.
Oh, really?
At that point, this was before the School of Bioengineering at Stanford. This was really early days, and so we were sort of pioneers of really combining engineering with biology and taking these 2 disciplines together. And in retrospect, it made a lot of sense just because the way I look at it is when you think about kind of the well-trodden fields of physics and chemistry and engineering and so on, there's a lot really smart people have sort of advanced those fields over the last, 100 years, multiple 100 years. And it's very hard to make more progress there versus if you look at the valleys between these fields, like bioengineering and so on, it's a lot easier to sort of move the
There's more white space for you to explore.
There's more white space, yeah. So it's these valleys that I think are really sort of fertile, sort of innovation ground. And obviously in hindsight, it made sense, but I mostly did it because I couldn't compete with like the really good electrical engineers.
Well, you've done all right. Hey, I want to actually explore that valley for a second. But we have a lot of different listeners here, entrepreneurs, executives, policymakers, and folks who, frankly, are probably in the academic field, whether they be doctors or bioengineers or, in other disciplines. I mean, obviously, in hindsight, it looks probably all makes sense when you look back at your career, what you did. But it's not easy to take that leap from being in a lab to like going and starting a company or even going to join a company. Like, what piece of advice would you give to someone who's thinking about doing that?
I think it's easy to overestimate the risk profile of making that jump of starting a company and so on. And so I actually, finished my PhD. I didn't know I wanted to start a company at that point. Said I had three routes ahead of me. I said I could go into academia, become a professor. And that seemed really interesting because all the professors at Stanford had one leg in a startup and so on. So that looked good. Then I said, maybe I'll just go join a large company and like learn the ropes and learn like product development and so on. And then the third was start a company around. And, in the end I said, okay, it's pretty hard to become a professor, but even if you do, it's kind of like a startup. You have to raise money, like write all these grants. And then you have the bureaucracy of a 20,000 person sort of like company. So it seemed kind of the worst of both worlds in some ways. Then I said, okay, if I start a company and it fails, at least I can always join the big company and my resume will say CEO. So I'll probably get a better job than if I joined the company right away. And so that's, in the end, if you can take the risk and, you're kind of, you have the financial sort of means to like take a year or 2 years to do a startup, I think it's much lower risk in reality in terms of what it means for the sort of continuity and the rest of one's career.
I love that. You're going to be CEO anyway, so if it doesn't work, you get a better job. That's awesome. Well, it obviously worked for you. What was for your first company? What was the insight, the unlock there that you guys that value that you guys explored and ultimately I think successfully because it was sold to Illumina pretty quickly, right?
Yeah. It was within 6 months of starting the company. So it was a crazy story. So I'll go into it. So the insight in my PhD was these machines that were sequencing DNA. The first genome in 2001 cost $3 billion to sequence. And so the idea was, obviously we'll probably want to sequence more than one genome in this world. We want to do it at something that's a lot cheaper. So the NIH sponsored all these programs. And so we won grants to get it down to $1 million and then $100,000 and then $1,000. And the idea was when you look at these machines, they actually use the same camera that's in the Hubble telescope. It's like I think it's like a $500,000 camera or $100,000 camera. The way sequencing works is you add one base at a time. The DNA is double-stranded, and you sort of build the complement one base at a time. And each time you add a base, you generate a flash of light. And so you need a camera to essentially look at that flash of light. And you're looking at, millions of different reactions simultaneously. The problem with these machines was they're using these really expensive sort of cooled cameras, but they had all these optics that were in between. So it turns out that if you put the reactions directly on the pixel of a sensor, rather than having all these optics where you lose 95% of the light, you could make do with a very cheap sensor that's like in your cell phone, or what's called a CMOS image sensor. And so that was really my dissertation was building a CMOS, image sensor. We used, we fabbed a chip at TSMC. And we built that, and we put DNA sequencing reactions on.
So the idea is lower cost and better sensitivity, essentially. I assume,
lower cost, and then the sensor you could throw away was disposable, so you just use it once a time, and you can miniaturize it because you could use semiconductors. You can, as transistors became smaller and smaller, you can put more and more pixels on, and so you could get the cost down like immensely. And so that was the idea for the first company. Sold that to Illumina, and it was a great experience at Illumina.
And that's a tough decision, right? It's like, did you guys know you had something at 6 months time point, or was it like, oh my gosh, we're so early, they're giving us an amazing, offer that we probably never expected, and this is a company that really can take this to the next level?
It was a tough decision. I think we thought we could build, a company that revolutionized DNA sequencing. Being 6 months into it was not an easy decision. I think the way I looked at it was, at the time, I think I was like in my late 20s, and venture capital in 2008 It was not venture capital today.
It was tough. It was tough, especially in your, in your field, especially in any field,
in
but
my
your
in
field,
my
yeah.
field, yeah, exactly.
Yeah.
So I could see that it wasn't like easy, especially anything that required, it was a very capital intensive business in terms of building and fabbing chips and so on. And I could see it was going to be like a really long road and probably pretty dilutive. Like at that point, we owned, the majority of the company, like 80% of the company between the two of us. I think the thing was like, look, I'm still in my late 20s. I could notch a win here on my belt in terms of an acquisition. Even if we, go and companies like a good exit at the time, like, amazing exit was probably like, 400 million, 500 million in that space. And so, you do the calculation of the dilution that you'd have, by the time you got there and the IRR in terms of how long it was. So it made sense to sell. And then I think in retrospect, we closed the deal in August of 2008. And we liked Illumina, so we were like, we're happy with stock. And
you didn't know what was going to go on with the market.
They were like, oh no, let's just do cash because it's easier for us and stuff. And we were like, okay, and so
sometimes better be lucky than good. Yeah,
exactly.
Yeah.
So in that respect, like we would have probably not been able to even raise like 2009.
Yeah, fascinating. Now, when you joined Illumina, was it like, okay, we're gonna go, this is our baby, we want to see this really be further developed, be commercialized, or did you kind of go to Illumina thinking, and frankly, a lot of founders think this, there's probably some milestone payments or handcuffs, I'm going to do that. I'm itching to start my next startup. Like, what was the mindset that you entered the big company?
Yeah, no. I mean, like we had like a 3-year earnout and, milestones and so on. So yeah, I was focused on the project, getting it out to market. I was still excited by it. I thought it was going to sort of revolutionize the field of DNA sequencing. When I joined Illumina, it probably cost a little over $1 million to sequence the genome. And,
wow.
and when I left, we reached $1,000 sort of cost of human genome sequencing. So it was a really amazing time in terms of like anything is possible. Stock was like ripping. It was really, I would say, maybe the glory years of Illumina. They went from a really small player in DNA sequencing to essentially 90% of DNA being sequenced on their machines now globally. I learned a lot during that period. Then, towards the end, we hit the $1,000 genome, and it just became something that didn't feel like it had the impact it once did, going from the $1,000 genome to the $500 genome to the $200 genome.
Why is that?
Just because, once you get to $1,000, whether it's 500 or 300, it's about the utility. What can you do with that sort of superpower of DNA sequencing? And, we were using advanced physics to get to the next level, really it was starting to get really, really hard to reduce the cost even further. And I'm like, do I want to rack my head in terms of solving all these tough problems on, lowering the cost by, sort of marginal amounts, or actually use that superpower of sequencing to do something clinically useful? And at the same time, I went through sort of a healthcare scare, and I sort of became much more involved with kind of how diagnosis happens, and sort of realized I think there's much more good that could be done by concentrating on that side of the fence rather than the research side of the picture.
Really interesting. You had worked and advanced the field of kind of the infrastructure layer, and now you wanted to move to the application later.
Yeah, exactly.
Yeah. So Guardant Health. So, I mean, it's amazing nowadays how, I guess I wouldn't say fully pervasive, but how far the field of liquid biopsy has come. And we'll talk about that journey that you guys have made. You've had some really interesting advancements recently. But for our listeners, I think people probably mostly know what liquid biopsy is, but talk a little bit about the biology, the work that you did to advance the field, and maybe compare it to the state of the art at the time. You founded Guardant Health in what year?
2012.
2012. So maybe talk a little bit about what it looked like then and what the insight was that you and your co-founder were working on.
Yeah, it's a really interesting story. This idea of liquid biopsy is this thing. It's mostly in cancer, prenatal, almost any disease where, the driving force of any cell is the DNA that's in the cells. So there's 37 trillion cells in the human body, and what turns out that some of the cells shed their contents into the bloodstream, including their DNA. This is called cell-free DNA. And so the idea is if you could take a tube of blood and you can sequence the hell out of that, what's called cell-free DNA in that tube of blood, you could potentially reconstruct essentially the genetics of disease or tissues that are in the body. And so it's almost like a catalog or an atlas of like everything that is going on in the body. And one of the diseases that is most prime for this idea of liquid biopsy is cancer, where when you think about cancer, it's fast-growing tissue, the tumor is growing really, really rapidly. But it turns out that the death rate and growth rate are kind of, somewhat aligned. And so as those cells are growing, a lot of them are shedding their DNA into the bloodstream. And so that is the insight and the promise is, rather than doing a surgical biopsy and finding where that tumor is, if I could take a tube of blood and essentially see that cancer genetically wherever it's growing in the body, but from a few teaspoons of blood, I can completely revolutionize the field. You can help late-stage patients, match them to therapy. You could detect recurrence in cancer survivors. And then obviously the most promising thing, exciting thing is really early detection of cancer. So you could have that blood test at an annual physical. It turns out that, this has been sort of like a dream of humanity for like 50 years or more.
Yeah. So Guardant now uses the very powerful technology that you guys developed in many different ways, right? It helps patients who have cancer match to the right therapies. That's one product line I understand. It obviously, if you have cancer and it's relapsed, it allows you to monitor. And then Holy Grail is now it's allowing you to sort of screen for cancer, right?
Exactly.
So, but that sounds great. That's taken a lot of years to get there, and that's created obviously a lot of the value in your company is that you're not just one single product. But I imagine as you reference, like even in the early 2010s, it was like high capex, not a lot of success in diagnostics in the venture back world. Like it's kind of been a graveyard. How did you guys as entrepreneurs? I'm sure that was your vision. I heard in a podcast you said that was always your vision from the beginning. But how did you choose where to start? Because if you tried to do all that, you would be dead in the water. So like, how did you sequence it? No pun intended.
Yeah, that's a great question. It was sort of like, I think the Tesla master plan in many ways, like, maybe before that, where, you look at some of the great companies out there, they figure out how to scale the price to performance sort of curve. And sort of sequence those events. What happens is most of healthcare has no leverage, which is why, it grows, which is why we spend $5 trillion now in healthcare in the U.S. and $15,000 per capita. I think they're selling like $50 billion of GLP-1s now in the U.S. like per year. And GLP-1 molecule doesn't get better. There's no like Wright's law or Moore's law in terms of like production getting to sort of better performance. But we realized this was different. Essentially, if we started with late-stage disease and we got really good at detecting stage 4 cancer in the blood, as we processed more samples, refined the processes, collected more data, that was going to be our compass to improving the performance to stage 3, to stage 2, stage 1, and essentially, open up successive S-curves. And so the first one is about 1 million late-stage patients in the U.S. It's now maybe a $5 to $10 billion sort of TAM. But then, as we improve the performance and could detect, earlier stage cancer in cancer survivors and recurrence, there are 18 million cancer survivors in the U.S., and that's about a $20 billion TAM. So essentially, the performance of the improvements of the first application sort of opened up the next market opportunity and the much bigger one. And then finally, early detection. There's 120 million Americans over the age of 45. That's a $50 to $100 billion opportunity. And so that, that was always the plan from the beginning. Start with the luxury market. When you think about late stage patients, they're costing the system, many hundreds of thousands of dollars. Obviously, it's very serious. It's acute care. And so that's a test you could also potentially charge more for because there's a lot more value in terms of the healthcare journey that you're in, and then essentially use that gross profit to essentially fund R&D. And so our view was, we call this technology compounding. Obviously compounding is the most powerful force in finance, but the same thing in technology as well. And so as we compounded, we increased the scale, and we essentially opened up these new markets. And so, obviously the details are very different in terms of how we actually achieved the performance, but the overall plan is the same from now as it was from 2012.
I love that. Just on the first product, it's a combination diagnostic therapy for late stage cancer for matching it. I understand.
It's called Guardant 360.
Guardant 360. Was part of the unlock there is that you actually were partnering with pharma, and they were they actually providing some, non-dilutive funding because it was a companion diagnostic, in other words?
Yeah. So the challenge with selling to the clinical markets is reimbursement takes a hell of a long time, right?
Oh, God,
and you don't get paid for a long time. I mean, it took us 9 years or 10 years to finally get like the last payer after we launched the test. So that, yeah, exactly right. So pharmaceuticals, I would say working with them was not necessarily non-dilutive funding, but, high gross margin sort of revenue that essentially helped us fund the clinical side of the business. And frankly, if you think about it, it's a spectrum where everything we're doing in the clinical side is sort of on the post-commercial drugs.
Mm-hmm.
And then working with pharma is sort of pre-commercial, the phase 1, phase 2, phase 3 trials, finding patients for those trials and helping them refine sort of the drug targets and so on. And so like even now the pharma business is large for us. We work with 200 different pharmaceutical companies on their trials and helping them with drug discovery and so on. And it's been a sort of nice leading indicator for us. It tells us sort of where the future of cancer care is going and gives us a little look ahead.
I said something earlier, and diagnostics has been I find it always to be a hard space and almost unfair to the innovators. And it's got all the challenges of a life science company, i.e. you have to get the technology to work, right? You have to do the animal studies, do the human studies, like actually write the papers, then you got to get it, prove that it works in the clinic, right? Then you got to, actually get reimbursement, to your point. And then you got to do all the commercial stuff that a traditional healthcare IT or services company does. So it's like you got to do it all. You got to raise funding, but I'm curious, what part of that journey was the hardest for you guys, did you think?
That's a great question. I don't wish my worst enemy to be in the diagnostics field. It's a hard industry because you have all of the burdens of all of healthcare with like the lowest gross margins. I would say the hardest part is reimbursement for the most part, because it's still very murky in medical device and in more importantly in pharmaceuticals and therapeutics. Reimbursement is much more a given. You see stocks pop once their phase 3 trials read out positively because essentially that that drug will be paid for. People can essentially model out what the TAMs are, what the potential like peak sales will be for that drug. That uncertainty on the diagnostic side is probably the worst thing for everyone involved, because you see companies launch diagnostics, but that doesn't mean they'll ever get paid for. People sort of like companies, tout like, oh, we got this reimbursement code, but even that doesn't mean they actually get paid on it. And so it's very, I would say, unclear in terms of like what the sort of economics are and what the business models are in diagnostic. And I think that does a disservice both to the companies, the startups, in terms of those trying to raise money, as well as investors who are deciding between different opportunities. And that's why I think having something with more clarity where maybe you do raise the bar to an FDA approval rather than what's called a sort of LDT. Or CLIA lab. But, as a carrot, you tie sort of like Medicare reimbursement if you're in certain like disease areas. And I actually think that would make this space a lot more investable than it is today.
Interesting. I was going to ask you, if you had a magic wand and you were a policymaker, what would you do to change it?
I think the other thing that's happening, which is exciting but also scary, is the shift from sort of intervention to data. And I think there's a lack of clarity or lack of understanding that it is better to measure twice and cut once than measure half a time and cut 3 times like we are doing in healthcare right now. But I think there's confusion, like as the shift happens to data and upfront like diagnostics versus just, sort of intervening blindly. There's a lack of understanding of the full health economic value of that. Like, people see, I think some of these tests, being more expensive or being more utilized, thinking that is something that is like when in reality, you're actually saving a lot of money. I'll give you an example. So in the UK, they adopted our test now for all lung cancer patients. Our test is more expensive than what they've been normally doing right now, just kind of small little tests and so on. Now they're doing our test up front. But what they found was because they're doing the test on a suspected diagnosis with a CT scan of these lung cancer patients, they're saving all this money in terms of these biopsies and other interventional sort of radiology things, and they're saving time. It used to take 4 months to get patients from sort of diagnosis to treatment. Now they're doing it in a few weeks. So they did an analysis, and the NHS found that by incorporating the technology, they're saving about 11 million pounds a year for those lung cancer patients that were under study and getting much better care because patients are getting on drugs for a few weeks. And so this is the thing is you're shifting the equation a little bit in terms of where you're putting your chips rather than putting thousands of chips on the drugs and interventions and some of them being misutilized. You put it up front and you do much better sort of interventions downstream.
Yeah, I mean, totally paradigm shift you're talking about exists in a lot of healthcare too, just preventative versus, intervention,
exactly.
right? And that's kind of what you're talking about. And by the way, I imagine this is going to be a challenge with Shield because Shield, you announced that's the product that's the screening product, correct?
Yeah, exactly. Yeah.
And you announced it about a year and a half ago. Is that when it was
we launched it in August of 2014 is when we got FDA approval.
2024, right?
Yeah, that's right. 2024. Yeah.
So, Shield, the TAM, as you noted, the number of people that could be affected, me, you, anybody would benefit from Shield, but it's going to lead to this paradigm exploration even more of how is the system going to how's this versus intervention. So I'm curious. Tell folks a little bit about Shield.
It's exciting. It's obviously the dream we had when we started the company. It was a blood test and an annual physical, and when I said we thought we were just going to sequence the sort of genetic code and sequence the hell out of it, deeper and deeper and deeper and reduce the noise and solve cancer, basically. But we hit a wall. We found out just looking at the changes in the 4 letters, the mutations, was not enough to get to the sensitivity required for stage 1 detection. And so we moved to sort of another sort of mark of DNA, which is called the epigenetics or methylation. So when you think about it, genome's 3 billion letters. There's 22,000 genes in it. Clearly, every one of your 37 trillion cells has the same DNA, has the same 3 billion letters or 6 billion letters, one from your mother and one from your father. The cells are very different. Your liver cells are different than your skin cells, different than your eye cells. Even though they have the exact same DNA, you sequence those cells, they look exactly the same. And it's because of these, what are called methylation marks or epigenetic marks. And so there are switches you can think about on those 22,000 genes. Think of the DNA as the hardware layer and the epigenetics as the software layer. So the hardware can do a bunch of things, but the software tells you what program's actually running right now in that cell and why it's behaving like that. The same thing for a disease cell or a cancer cell has a different software program. And so we developed a chemistry that allows us to see that software change or the software state or what's called methylation state in blood. And that got us to that next leg in terms of, even higher performance. It improved the sensitivity 100-fold over where we were before. And that sort of enabled Shield in terms of, getting to the performance required for early detection. And so, we started with colorectal cancer. It's the first liquid biopsy that's FDA approved for screening of cancer. And it's covered by Medicare. So there's about 50 million Americans who can get that test now at $0 sort of copay or near $0 copay.
And Helmy, is this I'm at the age where I've had my colonoscopy.
Yeah, sure.
All our listeners should get it. Is this a substitute for colonoscopy? Is this a substitute for Cologuard?
It has a first-line label, but we would never recommend anyone to not forego colonoscopy. Obviously, colonoscopy is the gold standard in terms of you can go in, it's interventional, you can cut polyps out and so on. Yeah. But I think the fact of the matter is there are 50 million Americans out of 120 million that are not up to date in screening today.
Yeah, I know.
There are 50,000 deaths from colorectal cancer a year. 76% of them are from those 50 million Americans who are sort of unscreened. And so the single biggest innovation and drop in sort of cancer rates in colorectal cancer we can sort of engender is by just getting everyone screened with Shield or one of the technologies out there. And the fact of the matter is stool testing, colonoscopy has been around for decades, and we still are stuck at, these 50 million Americans who are not screened. So clearly these are unpleasant and unpalatable options. And so we think now blood, if added to the mix, could make a huge, huge dent in terms of colorectal cancer death rates. So we estimate that this could be one of the biggest sort of like innovations in terms of actually dropping mortality from colorectal cancer, this blood test. We're seeing over 90% adherence to blood once it's ordered. 90% of patients actually follow through versus the existing methodologies have anywhere between 25% to 70%. And this is really exciting. And then the last thing is we just added, the ability to detect 9 more cancers with the test. And so we can now detect pancreatic cancer and lung cancer and ovarian cancer and so on. It's really just with a software update to the test. So it's really exciting. It really is this sort of precision medicine or data age of healthcare, where the more sort of like machine learning you can apply, the more conditions you can detect with the same test.
I assume you guys aren't just stopping at cancer, although that's where you focus to date. Like,
yeah.
Obviously there's certain things like cancer and rare disease and some other diseases where there's changes that are easier to detect. But like, are you going to be able to get into cardiology or other areas?
Yeah. So like, if you remember, the challenge with detecting the genome, and one of the sort of things that it maybe overpromised and underdelivered was, we got to the $1,000 genome. We thought, hallelujah, we're going to like have this like utopia of solving all diseases. And, what we ended up with is, I think really good progress in cancer, rare diseases, and infectious diseases, because those are the only diseases that change the genome.
Right.
Everything else only changes the epigenome or the methylation. So now that we developed this new technology, it gives us that bridge to be able to detect other conditions. And so that's what's exciting from the sort of machine learning AI side of things, is that as we test more patients now, we're seeing those signals in many other diseases, liver disease and cardiovascular disease and neurodegenerative disease, inflammatory diseases. And so what we're doing now is this sort of massive sort of machine learning like exercise, like as we sequence and test, hundreds of thousands, millions of like patients over the next few years. Fortunately, most people don't have cancer, 99% plus don't have cancer, but they may have other conditions. Some of them may be on Humira for rheumatoid arthritis or other things. We're getting all of that data, kind of de-identified, and then using that to essentially train the algorithms to detect all of these other conditions. So we think eventually Shield could be a sort of co-pilot to every primary care physician, where it's diagnosing hundreds of conditions, not just dozens of cancers.
I've noted that you and your co-founder actually are co-CEOs, and I think that's been for a while. That is not a very popular thing among investors generally, and not a common thing. Like, how did you guys? Was that always from the jump?
Initially I was CEO and he was president, so we kind of split the responsibilities. And then, if you think about it, people ask us like who our competition is, and because we had like 3 different areas, like, obviously the late stage stuff, the cancer survivor stuff, and early detection stuff, we basically had like multi-billion-dollar competitors in each one of those areas that were only focused on those areas. And so no matter how big we got, we always felt like an underdog. And when we were serious about early detection and we had to like, do these big trials and all this machinery, we realized that, superpower we had, or like an asset we had, was the fact that we had sort of 2 full stack founders here at this company. And so, a few years ago, we decided to, transform to this co-CEO structure. And we got some negativity from like, public investors and stuff. They said, and I think it's because they pattern match to cases where co-CEO is sort of like, kind of put on or forced on, some companies. For us, we've known each other since, 20 years and 25 years from the Stanford days. And any relationship, if there's a solid foundation of trust, then it works. If not, then it's not going to work. And I think that's the sort of like asset we had, is like, we know each other, we trust each other. And so it made sense, because it means we could go in these 2 different directions really, really fast, without sort of like, the added layer of like double checking everything and so on. It's worked out really well for us. I think
I love it. It's like form meets function almost.
Yeah, exactly.
Final question. You've obviously had an amazing entrepreneurial journey. if you were to impart like one lesson to aspiring entrepreneurs, like one thing that you like would be your piece of advice, what would it be?
There's a few. I mean, in healthcare, maybe talk about that a little bit more specifically. I think companies often focus on technological innovation, which is important, but I would say getting the business model right is probably 90% of the battle in healthcare. It's such a complicated field to navigate, all these entrenched incumbents that have a piece of the pie and are trying to force and push out like any innovators that come in. And so it really is David versus Goliath. And, you have to find, you have 5 smooth stones, find like the weak point in terms of entry. But it's really around like, how do I essentially monetize this innovation that I'm bringing in? If you figure that out, then I think you can solve like 90% of the problem.
Super, super great advice. Helmy, congratulations. Thank you for, it was a masterclass in diagnostics field and your journey, and just really appreciating you come on the Heart of Healthcare.
No, thank you for having me. It was a great discussion. Thanks.
All right. We're just